It's my pleasure to introduce Roland Wittler. Thank you again for your patience while we were setting up the YouTube stream. And I'm glad to introduce your talk, SANS serif, alignment-free, whole genome-based phylogenetic reconstruction.
Yeah, thank you very much. The short title of this talk is basically Sohn update, because I introduced Sohn itself 2 years ago at DSB in Dortmund. And today I want just briefly repeat what is Sohn, and then I will focus on new ideas and extensions on Sohn. In a nutshell, what is Sohn doing? We take a lot of genomes as input and then extract Kmers from the genomes to infer a phylogeny for these genomes. So usually the classical approach to infer a phylogeny is that you take some marker genes and you do a multiple alignment, or you align your genome to a reference to obtain SNPs. And then from this you infer pairwise distances, or you follow a maximum likelihood approach to obtain a tree, your phylogenetic tree. And in large-scale settings, this is of course difficult to do multiple alignments, or maybe you don't even have the annotations for your genomes. So there are also whole genome-based approaches to overcome this, and usually they are based on Kmers or on other similar patterns, but they all extract pairwise distances from the data to obtain a tree again. And the method Sohn is also a whole genome-based approach. It's also Kmer-based, but instead of inferring a tree, we infer phylogenetic splits. I will show you in a second what this means. And yeah, all in all, we don't need any annotations, we don't need marker genes, no alignment, no reference, and we don't even do a pairwise comparison in the end. And that's why we came up with the abbreviation Sohn, nothing. Without. So what are phylogenetic splits? That's a generalization of trees, and the idea is when evolution is tree-like and the data can be represented as a tree, you do get your tree, but there are also evolutionary mechanisms like horizontal gene transfer which cannot be represented as a tree. And here splits, phylogenetic splits, then show these non-tree areas of the data, and they also show the alternatives. Like in this example, we have the red split that separates the upper two genomes from the remaining genomes, and we have the green split that separates the right three genomes from all the others. And these two splits would not be compatible with the tree. So how do we get the phylogenetic splits from our genomes? So on the left, you see a visualization of 4 genomes: blue, green, orange, and brown. And they are visualized like a colored brown graph or string, any kind of string graph, whatever you like. The basic idea here is to show that there are segments of the genomes that are shared by the genomes, and there are segments where they differ. And the numbers here show, in this toy example, they show the length of these shared segments. So for instance, here we have a segment of length 2, and the length here is measured in Kmers. So we have 2 Kmers that we find in the orange and the brown genome, but not in the other genomes, blue and green. And we take this as a signal that brown and orange should be separated in the phylogeny from the other genomes. So in the phylogeny on the right here, you see this split of length or weight 2 that separates brown and orange from the other 2. And we also have 2 Kmers that are only in the blue genome, but not in the other 3, and 1 green Kmer, and so on. And we also have 3 Kmers, which are in the blue and the green genome, but not in all the other genomes. And this split can be found here. And now you see, of course, that well, we have, on the one hand, we have the split brown-orange versus the rest, and we have the split blue-green versus the rest, and of course they tell the same story, so we somehow want to combine these two splits, and we call them the inverse splits of each other. And the question is, how do we combine the two weights of these splits? Naive idea would be to sum them up or to take just the mean value of this, but in practice this doesn't work so well. Instead, we take the geometric mean. So why do we do this? On the left, you see some phylogeny. Let's assume we know the phylogeny and we pick one edge, and this edge separates the phylogeny into the red and the blue subtree. So we have an edge in the phylogeny. That means something must have happened here. So there must be some mutations, and these mutations lead to some sequences that, or some, the red variant of the sequence and the blue variant of the sequence. This means we have some Kmers, or let's say we have the red Kmers of that segment, and then we have the blue Kmers of that segment. And as a first proxy, we would also assume to have roughly the same amount of these two types of Kmers. And if we observe that we do not have the same amount, so we only would observe the one variant of a segment, but we don't have the inverse split, then this is a signal that something is weird. So this is why we take the product of the two weights, and this leads to a high weight only if both weights are high, and if one of the weights is low, this downweights the total weight completely. We can also add pseudo-counts in order to not lose any splits if one of the two splits would be 0. So that's the basic idea of Sohn. The first version, as I already said, was introduced in DSB 2 years ago, and meanwhile it's published and ripened, let's say. The first version was using a colored de Bruijn graph. In fact, the Bifrost data structure by Paul Milstead and Guillaume Mollet. And once the graph was built, we traversed the graph once, extracted all the unit ticks, and collected the Kmers, stored everything into a big trie to count the Kmers. Meanwhile, a student, Andreas Rempel, who will also present later on a different topic, re-implemented everything. He just used basically plain hash tables. So the new implementation does not have any dependencies at all. And surprisingly, this very simple data structure is way more space and time efficient than the original version. And he also included some filters of the splits. For instance, one filter is that from all the splits you get, you greedily extract those splits that are compatible to a tree if you really want to have a phylogenetic tree in the end. So since this was space and time efficient and it includes filters, we called this Sohn serif. That's the new abbreviation. How does it work in detail? The input are FASTA files. We scan the files, binarize the Kmers and store them into a hash table. And for each Kmer, we count in which genomes, in which input files this Kmer appears. So the first Kmer appears in genome 1, the second Kmer in the first 2, but not in all the others, and so on. In the next step, we have to combine those Kmers with the same occurrence pattern. And for this, we use second hash table. So we go through the first table and count how often do we have, for instance, this pattern, genome 1 and 2, but not all the other genomes. And at the same time, we also look for the inverse split. So we count how many Kmers do we have that appear not in genome 1 and 2, but in all the others. And these two numbers are then jointly stored in as one entry in the second hash table. And then from these counts, we compute the geometric mean, and then this weight well, if you visualize all the splits in a network like here, this weight then corresponds to the length of these edges here.
Sorry to interrupt you.
Yes.
May you please stop your screen sharing and start sharing again, because on YouTube they cannot see your slide.
Oh.
Which is weird, because instead they can see your face and mine. So please start screen sharing again.
Let me try.
At the beginning it worked, but then it stopped working. And yes, okay. So please go ahead. Thank you.
Okay, sorry. Yeah, so this is how we get the phylogeny. For an evaluation, we took a data set of about 3,000 assemblies of Salmonella subspecies. And here you see the runtime and memory. First, the runtime. Here you see the runtime of the original implementation with Bifrost and our tridata structure. And below you see the much faster variant, the serif re-implementation with the nicely slow linear increase of the runtime. And here you also see some numbers of other whole genome-based approaches, which take already for a subset of 250 assemblies, which is here. They already take hours or days, where serif now takes a few minutes. And also the memory usage has been improved a lot by these new data structures. One extension that we played with is UPAC characters. So sometimes the sequences contain Ns or other ambiguous UPAC characters, which is a bit annoying. And the default usually is, whenever you have a Kmer that overlaps such a UPAC character, you would just ignore it, or you skip that Kmer. And we wanted to try to incorporate these by considering all variants of that Kmer that this UPAC characters code for. Like you can see here, R can be an A or a G, and the Y could be a C or a T, and then we have like the Cartesian product of all the possibilities. Of course, only one of these four variants would be the true Kmer, and we have 3 false Kmers. But the idea was the true Kmer would support and would be supported from further Kmers that support the same split, and we would get rid of all the false Kmers downstream of the method anyway, due to the symmetry of the geometric mean, because we would not observe the inverse split. And it really worked out. So here you see precision and recall, an unrated precision and recall, which means we just count how many splits or how many edges in the tree are correct or wrong or missing. And the weighted precision and recall is also considering the weights of the edges. Original means we have simulated phylogeny and simulated sequences without any ambiguous UPAC characters, and then we get this precision and recall. And then we took the sequences and randomly inserted Ns every now and then. And the default version, where we skip all Kmers that overlap such a character, well, there we have a huge drop in precision and recall, as you can see here. But if we then instead replace all Kmers, all these Kmers by all the different variants, precision and recall go up again, and for the weighted precision and recall, we even get back to the original accuracy. Coding sequences. As I always mentioned, it's a whole genome approach, but nevertheless, we ask ourselves, well, what happens if we do have coding sequences at hand? If the genomes are annotated, why not using them? So a student of ours, Marco, implemented this. So instead of taking the whole genome, we only take coding sequences. And of course, this allows also to switch from the DNA alphabet to the amino acid version of the coded sequences. So Marco implemented this larger alphabet, and now you can feed Sohn with amino acid sequences as input, or you can also feed it with the coding sequences which are untranslated, and Sohn will do the translation then, and you can even select the genetic code that you want to use for the translation. So how does it perform in practice? Again, here we see recall and precision, in this case for some 90 Pseudomonas genomes. And here you see the accuracy for the whole genome version. So if you take the complete sequences, and then the question was, first, what happens if we restrict the input to the coding sequences without translation first? So we have less sequences, but maybe more meaningful sequences. So what do you guess? Do we get a better accuracy, yes or no? And well, the answer was it's worse. Yeah, and then the question was, okay, but what happens if we translate the coding sequences? What if we go to amino acid level? And there we really improve the results significantly. That was very nice to see. Yeah, finally, I want to show you a little application of the method. It's about metagenomics. So in metagenomics, you take a sample, and the sample contains many different genomes. You sequence your genomes, you have your reads, you can assemble these reads to contigs, and there are also binners which somehow assign the contigs which come from one genome into one bin, and these bins are then called metagenome assembled genomes, or MAG for short. You can also take several samples, and then what you can see here is it can happen that, or usually, of course, it happens that different samples contain the same genomes. So here, for instance, genome 3 is contained in both samples. And of course, you want to identify those genomes, but you don't know which MAGs. So you have different MAGs, but they actually come from the same genomes. And yeah, this is the setting here. We've given these many MAGs, and we want to cluster them such that all the MAGs in one cluster belong to the same genome. The idea was, let's try to use Sohn to infer a phylogeny, and then take this tree and chop the tree somehow into clusters. So we first computed the tree, and the tree looked like this. And well, on the first glance, you would say, wow, what a bad phylogeny is this? But the point is, we are actually not interested in a good phylogeny. We are interested in a tree that can be nicely decomposed into clusters. And this actually works quite well. If we zoom in, you see here one edge, and the subtree below this edge is almost composed of one genome only. The numbers here indicate the genome identifier. There's only one false positive here, which does not belong into this cluster. But if we move down, this different genome is separated in a different subtree. But we could also move further down, and then we also lose a true positive, which should belong to the same cluster. So the question is, where should we cut when we move down the tree? And by manually looking at this tree, I somehow came up with the rule of thumb to consider a subtree as a separate cluster whenever the current edge is longer than the edge above. That's basically it. The problem is the runtime and memory usage, because these datasets are usually really huge. And we investigated two different ways to improve this. Number one is a rank threshold. That means instead of considering all phylogenetic splits that we get, we restrict ourselves to some top ranking splits. So if we have n genomes as input, we say we only consider the top 50 times n many splits, or the 100 times n many splits. And of course, if we consider less splits, we can decrease the runtime tremendously. Another way to improve here is the usage of minimizers. That's something that Andreas experimented with. So instead of considering all the Kmers that we observe in the genomes, we take windows of a certain size, so and so many Kmers, and we only consider that Kmer that is the lexicographically smallest one within this window. And the larger we take the window, the fewer minimizers we end up with. This also saves time, but in particular it saves a lot of memory, as you can see here. And of course the question is, how far can we go with these parameters without losing too much accuracy? So we come to the results. You see the results for a data set. It's a simulated mouse gut metagenome from the Kami challenge. It's composed of almost 6,000 mags from about 700 genomes. And colleagues of us took care of the evaluation and compared this Sohn version to other methods. And as we can see, Sohn is with different parameters. It's the green and the reddish results here, and they end up here in the top right. So we have high recall and precision even compared to those methods, which are dedicated methods for this problem setting. So I come to my to a little outlook. There are further ideas we want to follow. We have another student, Fabian, who is working on multi-threading. We also want to try if it makes sense to, instead of just considering the occurrence of Kmers, to switch to counting the Kmers. And well, one question always for Kmer-based methods is which value of K should we take? So one bachelor thesis, Rebecca, she will work on 2 ideas how to estimate a good value for K. And we also want to investigate further some idea we already started with to measure the quality of the results without having a reference at hand, where we cannot compute the recall and precision. Yeah, with this, I conclude my talk and thank you very much.
Thank you very much. So I'm open to questions. Let's see the first one, which is, are there any ideas for exploring the accuracy drop from WG to CDS?
No, well, there are 2 factors. You have less sequence. Where are we? Let's see, right? You mean this? We have less sequence, but the sequence are maybe more meaningful than taking the whole genome. But the factor of having less sequence wins here. That's, well, more I cannot say. I don't know.
Okay. And I have one more question. This is of mine. Do you have any idea on the maximum number of genomes that you think you will be able to manage?
That depends on many factors. The length of the genomes, or in the end, the number of Kmers that you observe in the genomes, which you have to process. And also, yeah, and the number of Kmers depends on how similar the genomes are. The more similar they are, the more Kmers they share, so the less Kmers you have in total. And of course, I showed you these parameters which you might use to the heuristics that you can use to well, decrease runtime and memory.
Yeah, but I was wondering if it's feasible to analyze the coronavirus genomes. Which are, in this there are hundreds of thousands, but relatively short.
Yeah, they are really short. The Kmer set is small, so that should not be a big problem. The problem is that viruses are very short and they mutate a lot, so you have problems to find enough Kmers that are shared between the different viruses. So the method is not very well working on viruses. It is working, so you maybe do not get a very fine-graded phylogeny, but at least you get rough clusters. So you get a spider web network, but one corner is one subspecies or one variant and so on. But basically for viruses, for short virus sequences, there are better methods. Because on these short sequences, you can invest more effort to analyze them better than in this way.
Okay, thanks. We have a final question. How do you handle overlapping coding sequences?
So not at all. I mean, you read the Kmers from all of them, and if they're overlapping, you observe the Kmer twice. But so far, we're not counting the Kmers, so it doesn't matter if you observe it once or twice.
Okay. Thanks again.
Thank you.